Myself Sarvanshi Swaroop from School of Biochemical Engineering, IIT BHU Varanasi. I am going to present the study on spatial analysis of pioneer, settler and migrant transcription factor binding sites. This project was done under the guidance of Professor Shantar Ahmed, who is the Professor and Dean at the School of Computational and Integrative Sciences, Jawaharlal Nehru University, New Delhi. My mentor during the study was Ms. Manisha Kalsan, who is a PhD scholar at Skyway Lab, the School of Computational and Integrative Sciences, Jawaharlal Nehru University, New Delhi. As I mentioned in the beginning that it is the study of pioneer, settler and migrant transcription factor binding sites, so the question arises, what are these? Transcription factors are the proteins that bind to DNA to control transcription activity. Based on DNA accessibility patterns, transcription factors can be classified as pioneers. They have the ability to bind DNA target sites even in inaccessible regions and promote DNA accessibility. Settler transcription factors. They bind to the sites matching their DNA binding motif if these sites fall within accessible DNA. They do not bind to the inaccessible DNA. Last one is migrant transcription factors. They are very similar to settlers but are more selective. They bind only to a subset of their target sites even in accessible DNA. Their selectivity is driven by the interaction with additional cofactors. This study is intended to find where these three types of transcription factors bind with the mouse DNA. How closely do they bind to each other, or to find a pattern if any. It also aims to find what the genes in nearby locations of binding sites have functions, or which terms are more enriched in the nearby regions of binding sites. This study can be considered to consist mainly of three parts. First, preparation of merged datasets for pioneer, migrant and settler class of transcription factors. Second is the analysis of closest distance between these transcription factor binding sites. And the last one is functional enrichment analysis. In the first part, at the very beginning, we had files having binding sites for each transcription factor separately. A BD file contains binding sites information as columns for chromosome start and end, which means a BD file must contain at least 3 columns corresponding to the chromosome, its starting location and its ending location. For each class of transcription factor, the TF specific BD files were merged for all TFs belonging to one class to get merged dataset for pioneers, migrants and settlers. After merging the datasets, we get the data like this, having the location, the chromosome and the midpoint of the binding site and some scores. So after converting it into the BED, which contains the chromosome start and ending location, we extracted only the chromosome and one is subtracted from the midpoint of the binding site to get the starting location, and one is added to the midpoint of binding site to get the end position. This is the data of the migrant transcription factors, which contains around 9.5 lakh regions. Similarly, this is the data set for pioneer transcription factors, which contains around 13 lakh regions. This is the dataset for settler transcription factors, which contains around 2 lakh regions. All these tests were performed on the Galaxy server. After getting the dataset for all three transcription factors in BT format, the closest distance between transcription factor binding sites were analyzed. The merged datasets were used for prediction of closest binding site from other two datasets for each binding site in a dataset of one class. For example, the closest distance between pioneers were analyzed with migrants and with settlers too. This was done using the closest tool from BD tools on Galaxy server. Closest BD tool searches for overlapping features in binding sites A and B. In the event that no feature in B overlaps with A, it will report the closest distance, that is least genomic distance from A. It was operated on all 6 combinations, meaning if we have analyzed the closest distance between the pioneers and migrants, then we have also analyzed the closest distance between migrants and pioneers. After operating closest PD tool, the data looks somewhat like this, having the first 3 columns for the first type of transcription factor and the next 3 columns for the second type, and the 7th column contains the distance between these two strands. For example, this is the data between the migrants and the settlers. The first 3 columns contains the information about the migrants, the next 3 about the settlers, and the 7th column is the distance between these two type of transcription factors. To check whether the combination and its reverse yields the same result, the closest binding sites for both of these combinations were analyzed. After getting the results for the combination and its reverse, it is clearly seen that both are not the same. For example, the settler in the first region of both are same, but the migrant that binds closest to the settler is different from the settler that binds closest to the migrant, as the distance reported here are different. After getting the closest distance for all the 6 combinations, these data were sorted according to the distance. For analysis, the plots were made between the log of counts and the absolute distance in the interval range of 10, 100 and 500. For example, for the interval range of 10, we have counted how many of these binding sites lie between 0 and 10, then 10 to 20, and these counts were plotted according to the log of counts versus absolute distance. First plot is of the interval range 10 and second is of the interval range 100. And this plot is for the interval 500. All these plots were plotted using Python. From these plots, various results can be inferred. First, the number of counts decreases with the increasing distance, which can be clearly shown in these plots as all the lines obtained are the decreasing. With the increasing distance, the log of counts decreases for every combination. Second, the pioneers occupy a greater number of sites near migrants and settlers, but the number of counts decreases faster as they move to higher distances. In the plot, the red line corresponds to the pioneer versus settler and the green line corresponds to the pioneer versus migrants. These lines remain at the top for in the first plot and also in the second plot, but in the third plot it goes down at the distance more than 200 bases approximately. Next, the curve obtained for a combination and its reverse were different. Let's see in the plots that the blue line corresponds to the settlers versus pioneer and the red line corresponds to the pioneer versus settlers, which is the reverse combination of the first. And we obtain the different lines for both of these. The blue curve, which is for settler versus pioneer, and the pink curve, which is for settler versus migrants, were at the bottom except for some distance at the start in the plot of interval 10, which means for settlers, minimum number of pioneers and migrants bind closer to these at almost all the distances, and the counts were minimum for the closest distance in case of settler versus pioneers, which means minimum number of pioneers bind closest to the settlers at each interval, and maximum in case of pioneer versus migrants till 200 bases approximately, which means maximum number of pioneers bind closest to the settlers in almost each interval. Several more interpretations can be done from these plots. part is functional enrichment analysis. For the production of gene ontology terms, the tool Genomic Regions Enrichment of Annotations tool GREAT was used. GREAT calculates statistics by associating genomic regions with nearby genes and applies the gene annotations to the regions. As GREAT cannot take input of BAT having more than 5 lakh regions, the migrants and pioneers contain more than 5 lakh regions, so the overlapping regions were merged. But still, the migrants contains approx 8.5 lakh regions and pioneers contains approx 8.8 lakh regions, even after filtering the unique copies. So the maximum distance between regions to be merged was increased up to 50 bases, starting from 5 and increasing 5 at a time and checked at each stage. Finally, the maximum distance of 50, the migrants contain 4.9 lakh regions and pioneers contain 4.7 lakh regions approximately, which are now suitable for input in GREAT. Its output consists of tables for biological process, molecular function, and cellular components having values of several statistical parameters associated with them. This slide shows the parameters that are required for the input in GREAT tool. The species assembly, for this we have chosen mouse genome, the test regions which contains the BAT file of each transcription factor, and the background regions containing the whole genome, and by using the default association rule settings. This is the output of GREAT tool for the pioneers. The output contains the job description, several region gene association graphs, and the most important, the tables for biological process, molecular function, and cellular components. In the case of pioneers, there are 12 biological processes, and each table contains the term name, the binomial rank, the binomial p-value, and several other statistical parameters, and also the parameters for the hypergeometric test. Out of these, the parameters suitable for our analysis were selected. These were the binomial p-value, the hypergeometric p-value, and the total genes involved for each term. Similar process is done for the settlers and migrants also. From these tables, the functional enrichment plots were made between term name and the total genes, and the color of the bars depends on the p-value. The first plot is made using RStudio and the second plot is obtained from the Galaxy server. It is clearly seen that the response to dsRNA, the most of the genes are involved. In case of settlers, most of the genes are involved in DNA packaging. And in case of migrants, most of the genes were involved in chromatin silencing at rDNA. Similar plots were also made for the molecular functions and the cellular components. Many results can be obtained from these functional enrichment plots, but some of these are mentioned here. DNA replication, independent nucleosome assembly, and organization are the most significant biological processes in all the three types of transcription factors, which can be seen from the previous plots. Second, the maximum number of genes are involved in DNA packaging in case of settlers, chromatin silencing at rDNA in migrants, and response to dsRNA in pioneers in the category of biological process. NF-kappa B binding is the most significant molecular function in all three types of transcription factor, including several other common functions like sumotransferase activity, TAP binding, beta-2 microglobin binding, etc. So, this essay represents the overall spatial analysis of the binding sites of all these three transcription factors. Thank you.